Hello students, welcome back to my channel. Today my discussion topic is Hershey and Chase Bacteriophage Transduction experiment. In my previous two video lecture, you have already studied search for the genetic material, is it DNA or RNA or protein. In my previous two lectures, I have already discussed with you about the first remarkable experiment done by Griffith, that was Griffith transformation experiment to prove that DNA is genetic material. And after Griffith transformation experiment, AVERY, MACLEOD and MCCARTY again they were trying to prove biochemically that DNA is genetic material. They have almost proved that, but not of all scientific community was going to 100% satisfied to believe this fact that DNA is genetic material, because at that time it was a discussion about 1928 and after that 1930 to 1940 is a long time gap. There was not so defined process at that time. That's why DNA is genetic material. It has already been proved, but it was not clearly understood, not clearly acceptable by all scientific community. In 1953, scientists Alfred D. Hershey and Martha Chase, they were doing their experiment setup for a long period for proof that DNA is genetic material for a long time. Okay, and after all that experiment, they had published a paper in 1953 on about this experiment, that was Bacteriophage Transduction experiment. So prior to discussing that Bacteriophage Transduction experiment done by Hershey and Chase, which is widely or famously known as Hershey and Chase bacterial transduction experiment, we should have knowledge what's about bacteriophage. Bacteriophage, it is a virus. Bacteriophage, let's discuss something about bacteriophage. What is bacteriophage? Bacteriophage, it is a virus which attack bacteria, and it is also called phage virus. This one is also called phage virus. Bacteriophage, phage virus. As all of we know that virus is in position between, it is not living completely, it is not non-living completely. It occupies a middle portion. I want to say that it needs a host to show its living characteristics. Okay, until and unless virus get and host, they cannot show its living properties. Okay, so this bacteriophage virus, this attack bacteria. Okay, and let's have a look about bacteriophage structure. This is a rough structure of this is head, these are tail fibers, this is capsid layer, this layer is called capsid layer, and it has genetic material or DNA, and this one is known as base plate. So DNA is just encapsulated by its capsid layer. This is normal structure of a bacteriophage. Here phage virus. Okay, here Hershey and Chase, they used T2 phage, means T, it's a member of T even series. T even series, T means tadpole shaped, like tadpole shaped virus and T2-phage virus, and they used as host cell, as host cell is needed because without host cell the virus cannot show its living properties. So they used E. coli bacteria. E. coli means Escherichia coli bacteria. They have taken this Escherichia coli bacteria as they are easily culturable and easily maintainable in laboratories. And what's about this transduction experiment? What's about transduction? As in my previous lecture, when we had discussed about Griffith's experiment, I have already discussed with you about the 3 horizontal gene transfer process of bacteria. One was transformation, that I had already discussed in my that episode. Transformation wasn't just a feather touch. Transformation is nothing but uptaking of naked DNA. Uptaken of naked DNA from a culture medium. And what is transduction experiment? To insert a foreign DNA into bacteria by using phage virus, by using bacteriophage. T2 bacteriophage can reproduce within a living cell. T2 reproduces by invading these E. coli bacteria, and they are using the bacterium's molecular machinery to make more viruses. Prior to discussion about this transduction experiment, we should have a knowledge how this bacteriophage, they can invade Escherichia coli and use its molecular machinery. Let's draw. This is a bacterial cell and this is phage virus. This is bacterial cell. Say for example this is E. coli bacteria. This is E. coli bacteria and this is E. coli bacteria chromosome. This is bacterial chromosome or host chromosome. This one is phage attack, and phage inserts its DNA. This is phage DNA. What will happen next? What will happen next? Again this is bacterial cell. This one is phage, and this capsid layer, this is left outside, and this is known as phage host. It contains no genetic material now, and the enzyme encoded by this phage DNA, it can now break the host chromosome. Host chromosome is now going to break. Total host chromosome is breakdown of bacterial chromosome, and after that this one is, as all of we know, phage chromosome. What will happen next? There is no bacterial chromosome, is no more now. only phage chromosome. Only phage chromosome. They are all phage chromosomes. Here we can see from one phage chromosome a single phage chromosome, a single phage DNA. So here phage DNA replicates. And how can they replicate? By using bacterial materials and phage encoded enzymes. After that, slowly phage heads being assembled here. The spikes, they are being assembled here. What is assembled here? Phage head, tail, spike, all are assembled here. And here how phage genes are being assembled. Phage genes are expressed to produce other structural components of the phage. Progeniphages particle are being assembled. After that, the cell of this E. coli finally bursts of this cell and they come out. And here, as we can see, how they are just simply they are using this bacterial cell. This bacterial cell, E. coli bacterial cell, and finally the bacterial cell of E. coli, they bursts, E. coli cell bursts and they come out, and the progeniphages come out. Bacterial cell wall lysate, and this is lytic life cycle of a virulent phage such as T2. So, prior discussing this transduction experiment, this you should have cleared this concept first. Now, after studying the lytic cycle of phage virus, how can they attack a bacterial cell? After studying that, I have now Hershey and Chase experiment. Hershey and Chase, what they have noticed? They had noticed that T2 consisted only DNA and protein. And what was our debate? On search for the genetic material, is it DNA or RNA or protein? Hershey and Chase already know that T2 have only consists only DNA and protein. And what was their objective? Their objective was to clearly prove that DNA is genetic material. To prove this fact, they had used two radioactive isotopes. What was that two radioactive isotope? Phosphorus radioactive and radioactive. Sulfur and phosphorus. Clear? Why they used these two isotopes? Because they had that knowledge that DNA contain only phosphorus, as they contain phosphoric acid. Okay. DNA has phosphorus and DNA had phosphate, phosphorus, and protein contain sulfur. Protein contain no phosphorus and DNA contain no sulfur. Protein no P phosphorus and DNA means no sulfur. They have just used these two properties, these two unique properties about DNA and protein. And after that, what they have done? They have infected E. coli bacteria. Culture medium containing (32P) and culture medium containing 35S. Here in culture media they have grown E. coli. Here also they have grown E. coli. Here what is the scenario? E. coli took both phosphorus and sulfur. Here what is the condition of E. coli? Nucleic acid or DNA of E. coli contain all 32P, and here protein of E. coli contain 35S. Is it now very clear? And after that they use in this medium T2-phage. So what is that scenario now? As we have known from that lytic cycle that T2-phage use bacterial machinery. So here to construct progeny phages. So obviously T2-phage will use this protein, and here T2-phage will use this type of nucleic acid for making its own progeny phages. So here what we have get two set of T2-phages. Here T2-phage with protein radio labeled with this 35S, and here T2-phage with DNA or nucleic acid radio labeled with 32P. Two batch. Batch 1 and batch 2. Then what have they done? Again they have taken fresh E. coli bacteria. Fresh E. coli. fresh E. coli. Large T2 with 32P labeled DNA. T2-phage infect E. coli in 32P containing medium. E. coli, then lysis, progeny, phage with with 32P DNA. Here T2-phage infect E. coli with this T2-phage grow in 35S containing medium. E. coli again lysis and progeny phage with 35S protein. Labeled 35S labeled. After that, what will occur? Blending briefly. Lysate. Here also blending briefly. Radioactivity recovered in host and passed on to phage progeny. And here radioactivity recovered in phage hosts, not passed on to the progeny. Here radioactivity recovered in host and passed on to new phage progeny, okay. And here Radioactivity recovered in fudge ghost. Do you remember that? What about fudge ghost? Fudge ghost means after inserting its DNA, the empty capsid layer of T2 fudge is called fudge ghost, and this radioactivity recovered in fudge ghost not passed on to progeny. The new progeny does not contain any radioactivity. From this observation, what they had concluded, Hershey and Chase concluded that because it was DNA, not protein, which entered the cell by the presence of 32P and absence of 35S inside the bacterial cell. Immediately after that fudge had begun the infection process by injecting their genetic material inside the host. DNA must be the material for the function and reproduction of fudge T2. 32P but not 35S was found in the progeny fudges because the fudge genetic material inside the host would be partially repackaged in the progeny fudges being assembled during the infection process. Only genetic material, that is DNA, is passed from parent to offspring in fudge reproduction, and structural materials, that are proteins, are not. Besides this, I also want to share you with an information that Alfred Hershey shared the 1969 Nobel Prize in Physiology or Medicine for his discoveries concerning the genetic structure of viruses. So, from these 3 lectures on search for the genetic material, is it DNA or RNA or protein, in my first lecture we have already studied the Griffith's transformation experiment. In that experiment, DNA is genetic material was partially established, and after Griffith's transformation experiment, that was proved biochemically using different enzymes by AVERY, MACLEOD and MCCARTY. And that time also DNA is genetic material was established, and by this, after this experiment, Hershey and Chase's bacteriophage transduction experiment, it is now fully established that DNA is genetic material, not RNA, not protein. So be connected, and the next topic will be discussed about structure of DNA, which one is coming soon. So stay connected.